Chapter 22, The Lymphoid System and Immunity, Page 1. Overview. Maintain fluid balance, protection from infection and disease. Functions of the lymphatic system. A. Immunity. Filter fluid and ready immune cells. B. Lipid absorption. Lacteals absorb dietary lipids. C. Fluid recovery. Fluids reabsorbed into the blood. Lymphatic components. A. Lymphatic fluid. This moves from lymphatic vessels to veins. B. Lymphatic capillaries. These originate as pockets. Lymphatic capillaries are thin vessels with overlapping endothelial cells for one-way flow. C. Lymphatic vessels. Lymphatic vessels contain valves for a one-way flow, and they also have a beaded appearance. D. Lymphatic vessels. Lymphatic vessels are superficial and deep lymphatics from lymphatic trunks that drain into ducts. E. Lymphatic ducts. The right lymphatic duct. The right lymphatic duct collects lymph from the right side of the body above the diaphragm. It empties into the right subclavian vein. The thoracic duct. The thoracic duct collects lymph below the diaphragm and from the left side of the body. It empties into the left subclavian vein. Thoracic duct. The thoracic duct collects lymph below the diaphragm and from the left side of the body above the diaphragm and the rest of the body. The thoracic duct empties into the left subclavian vein. F. Lymphatic tissue. This is a region of dividing lymphocytes, germinal center with no fibrous capsule. Mucosa-associated lymphoid tissue, MALT. These are nodes associated with intestines. Tonsils. These are nodes associated with the pharynx. Page 2. Lymphatic organs. Lymph nodes. Lymph nodes are bean-shaped organs that filter lymph. Lymph moves from the cortex into the medulla and medullary cords. Afferent lymphatic vessels. These carry lymph from tissue to node. Efferent lymphatic vessels. These carry lymph from nodes to veins. Thymus gland. This is a pink, grainy organ behind the sternum. Within the thymus gland is the cortex with dividing and maturing T cells. Also within the thymus gland is the medulla with mature T cells that enter blood. These T cells respond to something foreign and not the body's own cells. Spleen. This is red lymphoid tissue that filters blood and stimulates B and T cells. Within the red pulp of the spleen are red blood cells. Within the white pulp of the spleen is lymph tissue with lymphocytes. Lymphocytes. Production occurs in bone marrow. Lymphopoiesis is the production of lymphocytes. 1. T lymphocytes. These mature in the thymus. T lymphocytes determine self versus non-self antigens. T lymphocytes include cytotoxic T cells and helper T cells. 2. B lymphocytes. B lymphocytes mature in bone. The production, lymphopoiesis, occurs in bone marrow. 3. Natural killer cells. These are not as specific as the others. They mature in the bone and they attack virus, infected, and cancer cells. Body defenses. A. Defense mechanisms. 1. Nonspecific defenses, immunity. This is also called innate immunity. Innate immunity does not distinguish between threats. Innate immunity is present at birth, and it is genetically determined. It can deny the entry or limit the spread of threats. A. Physical barriers. Skin and hair are physical barriers which prevent the entry of pathogens. Page 3. B. Phagocytes. Macrophages phagocytize debris. C. Immunological surveillance. This includes natural killer cells which destroy abnormal cells. The Golgi apparatus docks with the cell to be destroyed. D. Interferons. These are antiviral proteins that protect cells. E. Complement. These are proteins that aid antibodies in destroying pathogens. F. Inflammation. Swelling and redness isolates pathogens. G. Fever. Increased temperature increases phagocyte activity. 2. Specific defenses, or specific immunity. This protects against specific threats. It is coordinated by B and T cells. Specific immunity develops after antigen exposure. Active immunity is when the body is exposed to an antigen which produces a response. B. Classification of immunity. 1. Innate immunity. Nonspecific. It is genetically determined. 2. Adaptive immunity. Specific. This develops after antigen exposure. A. Active immunity. The body is exposed to an antigen which stimulates an immune response. Under active immunity, the body starts making a memory of the specific antigen. There are 2 types of active immunity: naturally acquired active immunity and induced active immunity. Naturally acquired active immunity is brought about by a natural exposure to pathogens. You get sick, but then you get better. In induced active immunity, there is an exposure to a specific antigen under controlled conditions. An example of induced active immunity would be a vaccine. Passive immunity. Passive immunity involves the transfer of antibodies from one source to another. An example of naturally acquired passive immunity would be maternal antibodies passed on to a baby. Induced passive immunity involves preformed antibodies given to fight an infection. An example of induced passive immunity would be antivenom. Page 4. C. Properties of immunity. Specificity to antigen, versatility to respond to many antigens, memory of exposure to antigen, tolerance of self-antigens. Cell-mediated immunity. A. Types of T cells. 1. Cytotoxic C8 T cells. These attack pathogens physically and chemically. 2. Helper CD4 T cells. These stimulate B and T cell response. B. Antigen presentation. Present antigen with major histocompatibility complex, MHC, which is a protein, at the cell membrane. This activates the T cell. C. MHC protein classes. 1. MHC class 1. These are in the membrane of all nucleated cells. They present cytoplasmic proteins on the cell surface. Abnormal protein will flag for death. 2. MHC class 2. These are found in the membranes of all antigen-presenting cells, such as macrophages and B cells. APCs internalize pathogen, present antigen on cell surface to activate the T cells to watch out for that antigen. Antigen recognition. T cells recognize class 1 or class 2 plus antigen with T cell receptor. If the T cell comes into contact with MHC and the antigen for which it is specific, activation occurs. The CD co-receptors help to dock. CD8 co-receptors on cytotoxic T cells. These aid in binding class 1 MHC plus antigen. CD4 co-receptor cells on helper T cells. These aid in binding class 2 MHC plus antigen. Page 5. Activation of CD8 T cells. 1. Classes of CD8 cells. A. Cytotoxic T cells. T cell receptor and CD8 co-receptor contact class 1 MHC plus antigen. This targets the cell, and the cell is destroyed via perforins. B. Memory cytotoxic T cells. Once produced, these remain active. Antigen re-exposure causes rapid cytotoxic cell differentiation. Activation of CD4 T cells. 1. Classes of CD4 cells. A. Helper T cells. T cell receptor and CD4 co-receptor contacts class 2 MHC plus antigen. Cytokines stimulate T cell division and B cell activation. They link to the B cells to help fight the antigen. B. Memory helper T cells. Once produced, they also remain active. Antigen re-exposure causes rapid helper T cell differentiation. I have a note here that cytokines are cell signaling chemicals. Antibody-mediated immunity. B cell sensitization and activation. Naive B cells with membrane-bound antibodies bind antigen. Sensitized B cells present antigen with MHC class 2, such as macrophages. Previously activated helper T cells can bind B cell and activate it. When this occurs, the B cell becomes a plasma cell antibody producing powerhouse. Classes of B cells. A. Plasma cells. Plasma cells produce antigen-specific antibodies. B. Memory B cells. These two, once produced, remain active. Antigen re-exposure causes rapid plasma cell differentiation. Antibodies. Heavy and light chains with variable regions bind to antigens, and constant regions They have 4 chains. They can neutralize, agglutinate, opsonize, and activate or complement macrophages. Opsonize means to coat with antibodies so the macrophage can phagocytize the pathogen without difficulty.